This method can help answer key questions in the pulmonary care field, such as why do patients who survive hospital-acquired pneumonia have such poor long-term outcomes? The advantage of this method is that it's simple and reliable, which means that anybody who comes in the lab can be taught it, and the next day they're generating useful and repeatable data.
Though these analytical methods can be applied to provide insight into the infection of cultured cells in vitro, they can also be applied to animal models and human patients.
To produce cytotoxic supernatant, wash a 150 centimeter dish of endothelial cells with HBSS and dilute a P. aeruginosa colony to an absorbance of 0.25 at 540 nanometers. Further dilute the bacterial cells to allow a multiplicity of infection of 20 to 1 in 20 milliliters of HBSS, and seed the bacteria onto the plate of endothelial cells. Then place the infected cells in a 37 degrees Celsius and 5% CO2 incubator for 4 to 5 hours.
It is essential that the incubation of the bacteria and the endothelial cells occur for an appropriate length of time. If the incubation is too short, the amyloids will not be released into the supernatant. If the incubation is too long, the cells will actually lyse and release all of their contents into the supernatant. The indicator that we use for an appropriate duration of incubation is the formation of gaps in the endothelial monolayer.
When gaps can be observed in the cell monolayer by light microscopy, harvest the supernatant for centrifugation to remove any cellular debris.